Okay, I'm going to be recording the Neuro PowerPoint. It's really kind of an A&P assessment part, but you've really got to understand this to understand some of the neurological disorders. Otherwise, I have posted the PEDS neurological disorders in a outline format from my notes and a review of the chapter. You only have to know what's in that PowerPoint for exam purposes or in that outline. I know the book covers a few extra things, but you don't have to know those. Also, you want to have your PEDS book and your MedSearch book along with the PowerPoint that I put up for this lecture to go along with me. All right, so I'm going to get it ready. Okay, let's talk about the neurological system in general. And a lot of stuff that we talked about in health assessment the first semester is going to come back to haunt you. The nervous system cells. There are two types of nervous system cells. There are the neurons and the neuroglia. And you really have to understand these to understand what's going on. Characteristics of the neurons. We'll start with the neurons first. Well, they're specialists. What do they specialize in? Impulse conduction. And they're classified according to two types of criteria. So if you were to ask me what do they specialize in, I would have to say two things. They specialize in excitability and conductivity. Excitability and conductivity. The first type of criterion is the direction in which they conduct impulses. Well, that can be afferent, efferent, or interneurons. Afferent are your sensory. These neurons transmit impulses, of course, towards the spinal cord or brain. They facilitate sensation. The efferent are the motor neurons that transmit impulses from the brain and spinal cord toward muscle. These facilitate, of course, movement. And then your interneurons are connected, conduct impulses between your sensory and motor neurons. The second criteria is the number of processes they have. You can have multipolar, bipolar, and unipolar neurons. The multipolar neurons have only one axon and several dendrites. There's a typical picture of a neuron on page 906 of your MedSearch book, figure 43-1. I also have given you a blank neuron to label with the pediatric neurological disorders, and here is a cheat sheet if you needed it. You can go by that, and we'll go over each one of these processes. But you can see here a multipolar, one axon, one dendrite with several dendrites, and that again is this typical picture. Now we have bipolar ones, and thank goodness it's not the bipolar that we think of. Bipolar neurons that have only one axon and one dendrite. So you would come out here and where you see the dendrites coming off the cell body, you would only have one. Then you have the unipolar, and these are kind of our deformed neurons. They start out that they're going to be a bipolar, a 1 and a 1, but somewhere along the line they fuse together and don't separate so that they really are not functional. All right, so just remember the structure of a neuron. All neurons have a cell body and at least two processes, one axon and one or more dendrites. Let's talk about each part. The axon is a single process that extends out from the cell body and terminates in branched filaments, and you can see that on page 906. It conducts impulses away from the cell body. Now, it can be myelinated or unmyelinated. Of course, myelinated means that it has a sheath, and the sheath is composed of a white lipid substance that helps insulate the conduction of those impulses. Now that's why we have babies that are not born walking and sitting up and rolling over, because it takes a while for those to myelinate. So that's the reason it takes a while for myelinization to occur. Now, if you'll also note, there's small gaps in the myelin sheets, and those are called the nodes of Ranvier. At the end of each axon, there's a terminal area known as the synaptic knob or terminal. And you can see there, that's where we come out into those finger-like branches there. And they contain neurotransmitters. And typical neurotransmitters have either excitatory or inhibitory influences. Excitatory influences would be like those of acetylcholine, norepinephrine, serotonin, dopamine, histamine, some of those. Some of your ones with inhibiting would be like your GABA, G-A-B-A is a big one, your endorphins. So those are some typical examples. Oh, what are the neurotransmitters? I just went over those. Those with excitatory or inhibitory influences. Excitatory would be like acetylcholine, norepinephrine, serotonin, dopamine, histamine. Those with inhibitory influence would be like your GABA, GABA, or your endorphins. All right, so what else affects neuron transmission besides neurotransmitters? Well, lots of things can do that. The distance between the actual impulses or neurons can matter. The distance, because if it's too long between them and you don't have those interneurons functioning, then you're not going to get a full impulse. The strength of the impulse. The strength of the impulse. And part of that's based upon age, because as you get older, acetylcholine just don't do what it used to do in the time that it used to do it. A lack of oxygen could cause or affect neuron transmitters, like anesthesia. If you're out under anesthesia, we don't want you moving, right? Changes in pH. We know that acidosis depresses the central nervous system, and we know alkalosis excites the central nervous system. Some of your drugs, caffeine, theophylline, we know those affect excitability. So there's several things that can affect transmission besides the actual neurotransmitters. So what is the relationship between sodium, potassium, and chloride in the role of transmission? If you look there on page 906, let me get my notes here straightened up. If you look down at that figure 43-1 and you picture along the outside of that axon, if you were to draw positives all along the axon, because it's positively charged on the outside, that's also where you have sodium, which is positively charged, and chloride, which is negative charged, to keep it in balance. Also on the outside, running along the outside of the axon, is calcium, Ca, and it's also positive charged. And calcium is known as the gatekeeper. Inside the actual cell is potassium. Inside is potassium. And inside the axon, or the neuron itself, is usually negative charged. Now, if you'll remember back in A&P, this is called a resting membrane potential. Then you have a stimulus that comes along into the dendrites that stimulates the cell body that runs down the axon. As that stimulus starts to stimulate, the gatekeeper, calcium, will open up. And when it opens up, sodium's going to go in and potassium's going to go out. Sodium's going to go in, potassium's going to go out. You're depolarizing. You're getting ready for the action potential to occur. Once that impulse is carried to the adjacent cell, releasing the neurotransmitters, then that's called the action potential that has occurred. And you have to have sufficient strength for that action potential to occur. Once your action potential occurs, then you have repolarization, where calcium opens back up and the sodium comes back out and the potassium goes back in, so that you go back to a resting membrane potential once again. And that was in a nutshell. The dendrites. The dendrites are also a part. They're the tiny tree-like areas that are distal ends of the dendrites. They have sensory neurons called receptors, because they receive the stimulus that start this impulse conduction. We start life with about 100 billion of those, but depending upon what lifestyle choices we make, we can kill those off. Characteristics of the neuroglia cells in comparison to the neurons is they do not conduct impulses. They simply support, nourish, and protect the neurons. So they're kind of referred to as the nerve glue. They do vary in size and shape, and they're about 5 to 10 more times common than your neurons. There are 4 major types of neuroglia cells. There's a brief discussion on page 906. There are the astroglias or astrocytes. Astrocytomas are the #1 brain tumor, so we know where they originate, right here. They're the star shape that provide physical support for the neurons and regulate the chemical environment. By regulate the chemical environment, I mean they're part of the blood-brain barrier. And if you'll remember, the function of the blood-brain barrier is to maintain homeostasis of the brain through selective permeability. To maintain the homeostasis of the brain through selective permeability. Because normally there's just certain substances that enter by way of the capillaries into the cerebral spinal fluid. The barriers protect and is only permeable to certain things and letting certain medications, for example, go through. You got the oligodendrocytes that form part of the myelin sheath. You've got the epididymal cells that form part of the blood-brain barrier as well and help regulate the composition of cerebral spinal fluid. Then you got the microglia, which appear to be phagocytic in function. They're usually small stationary cells, but if you have a lot of inflammation or degeneration in the brain tissue, they enlarge, move about, and engulf or phagocyze, picking up the debris. Okay, let's talk about the nervous system divisions. We know that there are two major divisions. There's the peripheral nervous system and the central nervous system. Peripheral and central. Just a chart to put it all together. You've got your nervous system. The central nervous system is further broken down into the brain and the spinal cord. Here's where we get into trouble with neuroseptic defects if that neural system does not develop and things do not close well. We have neural tube defects. The peripheral nervous system is broken down into the afferent and the efferent. The efferent is further broken down into the autonomic and somatic, and the autonomic is further broken down into sympathetic and parasympathetic. As far as the peripheral nervous system, that's where our cranial nerves are located. 31 pairs of those that carry impulses to and from the spinal cord. And again, it's further divided into the somatic and autonomic. Now, the somatic supplies nerves to our skeletal muscles, and the autonomic regulates our day-to-day things like our cardiovascular, respiratory, endocrine system. Then that is further broken down into the sympathetic and the parasympathetic. So what's the difference between the sympathetic and the parasympathetic? The sympathetic functions, of course, to maintain homeostasis or balance. It can increase its activity when necessary for any type of need. And typical responses you would see would be to stress, to include things like, number one, pupils dilate. Why do they dilate? They're looking for a way out of the situation. It's kind of like an adrenaline rush. The heart rate and blood pressure increase. The bronchioles dilate. You start breathing heavier. You start sighing. And the anal sphincter slams shut. So that's your typical response that you would see with the sympathetic response. Pupils dilate, heart rate increases, blood pressure increases, bronchioles dilate, and the anal sphincter slams shut. The parasympathetic conserves the body's resources. This is our day-to-day control of like respiration, GI, bladder, things like those things that keep you going. You got to eat every day, your feeding activities, your circulation, your elimination, all of those are included. All right, the central nervous system then is composed of the brain and the spinal cord. The brain is full of bony coverings. The bony coverings include the vertebral column and is encased by the spinal cord and the cranial bones. Now, when I say bony, we're bony. There's 8 bones of the cranium of the brain, and there's 14 facial bones. Then your spinal cord is encased by 33 vertebrae. 7 cervical, 12 thoracic, 5 lumbar. 5 that are fused to form the sacral, and 4 that are fused to form the coccyx. So we're bony. That's all I know to tell you. We're bony. Then you've got the membranous layers, the 3 layers of the meninges, and your Pete's book happens to have a good picture of that on page 928 at the bottom. You can see there, it shows you the typical section of the top of the head, showing the meningeal layers. You've got the first one, the dura mater, which is what I call the tough mother. That's the white, fibrous, most outer layer, and it's there to protect. Then you've got the acronoid membrane, and this is your cobwebby, delicate membrane. It's the middle layer, and that's where your cerebral spinal fluid is produced. It's the delicate middle layer where cerebral spinal fluid is produced, cobwebby. Then you got your pia mater, or your pia mater, however you say it. It's your tender, sweet mother. It's the transparent layer that's directly attached to the surface of the cord and the brain. So it contains your blood vessels, your nutritive layer. It's what you would think of a mother doing, feeding it, nourishing it, and help protecting it. Your cerebral spinal fluid provides protective cushion around and within the brain and spinal cord. We know it's clear, colorless. It has a specific gravity of 1.007. And a typical amount of cerebral spinal fluid would be 135 to 150 mils. Now, the spinal cord itself is a downward continuation of the medulla oblongata. It starts at the level of the foramen magnum, which is the hole at the base of the cranium, and ends at L2. So this is typically about 17 to 18 inches long in the average body. And it is also the site of reflex pathways. Now, there are 6 structures of the brain, and this is just a review. The cerebrum, which is the largest part of the brain, has 2 hemispheres, we know, the right and the left being the dominant. And it's also known as what we call contralateral. That means it controls the muscles on the right side, so affecting the opposite side. You can see here your right hemisphere mostly is your non-speech, your melodies, your crying, your laughing, all of those things. The right and left hemisphere is further divided into 4 lobes, your frontal, parietal, temporal, and occipital. And you can read through these to see what we've done. Now, parietal lobes, a lot of things that we talked about is affected in that. The only thing I'll mention for now, and we'll come back to some of these later when we talk about the actual assessment part, is graphothesis and steronogenesis. If you'll remember, graphothesis, graph, you're drawing, so you have someone close their eyes and you draw like the figure 8 in the palm of their hand. If they cannot identify the figure that you draw, then you put an A in front of that, A graphothesis. Steronogenesis is when they have the ability to feel an object and tell you what it is with their eyes closed. So you would have them close their eyes and put like a pencil or a penny or a key in their hand and have them identify it. If they can identify it, then that's intact. If not, then they have A steronogenesis. Okay, the next part is the diencephalon, and this is lies on top of the brainstem, and it has 2 major components. You have your hypothalamus and your thalamus. Your hypothalamus controls or affects appetite, temperature, mine's doing overtime on the appetite, water balance, pituitary, emotions. Your thalamus acts as the relay point of your sensory tract, except the olfactory nerve, and it helps play a role in awareness of pain and alertness. Your cerebellum is located at the base of the brain, and this is just below the occipital lobes. Now, when you think of that, you should think of equilibrium, balance, so that someone who has damage to this part of their brain results in what we call cerebellar ataxia, and they will not be able to be steady on their feet. The brainstem, which we call the bulb, connects the brain and the spinal cord and has 3 parts, the midbrain, the pons, and the medulla oblongata. The main thing you need to remember, some of your reflexes, but the big thing is your respiratory center is located in that area. So you have someone born with damage to the brainstem or the brainstem not being intact, then they are also not going to be able to breathe. The limbic system is the 5th lobe, and it's wrapped around the top of the brainstem and buried deep within the temporal and the frontal lobes. You can see there are several different emotions: anger, sadness, happiness. So your emotions are strictly tied to the limbic area. It's essential for normal memory. Oops, just kidding. Some of you are in trouble, it says. But sometimes we do get into overload, and that does cause some issues. And the last and final structure of the brain is called the reticular activating system. And this is the gatekeeper of consciousness. This is what controls our sleep-wake cycle. And as you can see here, I've got a little cute little monster here for us, so that if things don't go well with sleep-wake, sometimes we feel like this. Sometimes I even look like that. All right, that's the first section of that part. And I will be narrating the next section shortly.